Okay, so yesterday we have discussed about the structure of the muscle, and so that it consists also of electrical gap junctions between the muscular cells, and then it is connected. All of this muscle cells group is connected to a motoric neuron, yeah, which has this varicosities, which can transfer transmitter through the vesicles to the surrounding or to the neighboring muscular cells, yeah, to excite the muscles motion. So these are the actual microscope image, yeah, from the muscles. These are the mitochondria, the band one, and the sarcomere, and the intercellular space here is this the gap between the two muscular cells. So if we make model illustration of this in microscope image, it looks somehow like the picture in the bottom part. So as we have also mentioned yesterday, that muscular function started from the nanoscale level. Yeah, even at the molecular level, the tropomyosin move, yeah, along the myosin, yeah, or vice versa. So it has this actin, and then there is a binding site. So this will move along this myosin head, yeah, in the form of sliding mechanical motion. Sliding filaments as the basis for mechanical power stroke and contraction of the muscle cells. This is the cock myosin heads. This is the myosin heads. Binds to the actin, establishing a bridge before sliding. Compared with this, so the myosin will be sliding along the actin, yeah, along the actin to make the contraction. So the myosin head become flex, which causes the actin filaments here to slide to the right. So the whole actin slides because the myosin head flex. So this is how the signal will look like in the muscular cell contraction if we measure using electrodes. So the graphs show contraction of muscle cells in response to a train of action potential in the axon terminal. So the upper part shows the axon terminal of the motoric neuron, that give excitation to the muscle cells. So this is the innervating action potential, which have varying instantaneous frequency, varying firing rate. So in this 1, 2, 3, the frequency is very low. Yeah, the frequency is very low. So you see that at the moment that here there's a spike or a pulse in the axon, it will cause the muscles to contract by one time. Yeah, there will be a twitch. When there are two pulses here at the motoric neuron, at the axon of the motoric neuron, then it will stimulate or initiate two twitches of the muscular cells. Yeah, with a longer period compared to the only one twitch. The one twitch will last about 100 milliseconds. So here you see it looks similar like the previous discussion on neuromuscular a neural cell, yeah, the shape of the muscle. When you give an impulse, then there is corresponding voltage, looks like that. When its action potential triggers a delayed contraction of muscle cell known as muscle twitch. The successive twitches interfere with each other, so they are mixed with each other, and produce a prolonged and sustained muscle contraction known as muscle tetanus. So maybe you have heard about this one disease caused, I think, by bacteria, Titanus disease. So where the muscle is contracting very quick at very short interval. So because of the high firing rate frequency, because of the high firing rate frequency like this, and when it is like this, then they interfere, they are overlapping each other, then will cause a prolonged muscular contraction like this. So this is what occurs in people who has tetanus, and possibly also people who have other disease which cause them to have uncontrolled muscular contraction, because the motoric neuron fires signals at this uncontrollable, very high frequency rate into the muscular cells, and to produce this long prolonged twitches, which makes them a very big contraction like this. Okay, so that is the end of the muscle discussion. Then let me. Then we will continue with the next discussion on the heart. Yeah. On the heart. Yeah. Okay. So we have learned previously about cells. Yeah. To review again. In the last week, we have started the discussion on cell. Yesterday, we continued discussion about the cell morphology, structure, and physiology. And then we started to discuss about neurons and the receptors, the sensory receptors. Then we had discussed muscles, continued until this morning. So now we study a very important organ within our body, which is the heart, basically, where many of the electromechanical functions are embedded within it. Yeah. So we'll study first the structure and then the function. The heart is the motor for the circulatory system. It pumps blood through the systemic and pulmonary circulation loop. So in the biomedical instrumentation, maybe we have reviewed, you haven't got in the introduction to biomedical engineering, maybe we have reviewed a little bit about systemic. Systemic is the systemic circulation loop, is the blood circulation from the heart towards the rest of the body, except the lung. While pulmonary circulation is the systemic circulation loops distribute oxygen-rich blood to give oxygen to each of the cells in the body, so for the metabolism, and then taking back again the CO2 from the somatic cells. The pulmonary circulation loops, on the other hand, carries the carbon dioxide or carbon dioxide-rich blood into the lung to be circulated back to the atmosphere and then capture the fresh air, oxygen from the air through the lung. So the heart receives blood from the pulmonary and systemic veins and pumps it into the systemic and pulmonary arteries respectively. The heart is a muscular organ, that's why it is also basically works similar like the muscles, only it has more rhythm compared to the other muscles. And also the other muscle works voluntarily with our consciousness. The heart works involuntary muscle without our consciousness controlling it. So with rhythmic contraction, force blood to circulate within blood circulation loop. The heart represents a major source of numerous biosignals. So basically, if we consider again ECG, the life sign, the fetal life sign basically comes from the heart. Blood pressure also will originate from the heart pumping. What else? Uh, many others. Yeah, I think even other biosignals, uh, biochemical biosignals are also related with the blood circulation. Body temperature also is related with the blood pumping from the heart. So that is why the heart can be considered as a major source of this numerous biosignal. So if you consider the structure of the heart, it can be considered as a double pump with four chamber here because it pumps into the two different circulation loops. The first one is the pulmonary loops into the lung, and the other one is to the rest of the body or the systemic circulation loop. So like this, it consists of the right and left atria. Atria is the upper part of the upper part chambers of the heart, the right and the left, and the right and the left ventricles. Okay, so the right atrium receives blood from the vein, yeah, and it has this tricuspid bus. So this is the first pump, yeah, on the right side of the heart, the right atrium and the right ventricle. The blood from the systemic circulation enters the vein into the right atrium, then goes to the right ventricle before being pumped into the arteries, through the arteries into the lung, yeah, through this pulmonary valve. Then blood from the lung containing the oxygen-rich blood, the vessel from the lung, the veins from the lung containing the oxygen-rich blood will enter into the left atrium, yeah, of the heart, then goes to this mitral valve before being pumped to the rest of the body in the systemic circulation loop through this left ventricle via the aortic valve. Yeah. In between the two ventricles, there is this interventricular septum. So this is the model, and this is the actual illustration of actual anatomy of the heart. Yeah. So this is the systemic vein where the oxygen poor blood or carbon dioxide rich blood enters the heart from the rest of the body, from the systemic circulation. Then uh, from the right atrium, it goes into through this tricuspid valve, and will open and goes to the right ventricle before being pumped into the pulmonary valve, into the pulmonary valve here, goes here, and, via this pulmonary valve into the pulmonary artery, and will go to the right and left lung, where then the blood will exchange the gas through the lung in the surrounding atmosphere. The carbon dioxide will be diffused out and then exhaled out from our body to the surrounding atmosphere, and we inhale in the oxygen and diffuse that oxygen into the blood in the lung. Then that oxygen-rich blood will go again through the pulmonary vein to the left atrium, yeah, on the left side, upper left side, then going into the left ventricle through this mitral valve, yeah, and before being pumped into the aortic valve, yeah, aortic valve, through the aortic valve, through the aorta, and then being distributed throughout the whole body via the systemic circulation loop. Okay, so this is something which you as biomedical engineer needs to learn and understand. Where do they come from? Yeah? Otherwise, yeah, because again, the heart is a major source of biomedical signal or biosignals. So when we consider this, a lot of the pumping actions are basically done by the valve. Atria. The atria functions to prepare the heart to be the blood to be pumped. Then the pumping is done to when it goes into the valves, into the atrium. The atrium goes into the ventricle, the bottom part. Then it is being pumped by the ventricle into the big arteries, either pulmonary arteries for the pulmonary circulation or the aorta for the systemic circulation. Okay, so yeah, therefore the lower part here is usually thicker, has thicker muscles, muscular tissue compared to the upper part, because the lower part is the one that conducts the pumping function. So the valve between the atrium and ventricle functions to make the distribution of the fluid only in one way. The blood can only flow from the atrium, the upper chamber of the heart, into the ventricle, the lower chamber of the heart. So something like this. So the structure is like this. This is the valve, and it is connected with a cord-like tendons here, and connected to a papillary muscles. So here, if you see the structure, blood can flow only downward. Afterwards, it will close. Then blood cannot go to upward direction, cannot go in the reverse direction. It has to go to the other valve towards the arteries from the chamber. Yeah. So here, for example, when the valve is closed, the blood is contained within the atrium, like this. And when the valve is open, blood flows from the atrium into the ventricle. So this is atrioventricular valve. There are two types of them. The one on the right side, yeah, for the pulmonary circulation loop. Yeah, the valve between the right atrium and to the right ventricle is called the tricuspid valve. Yeah, why is it called tricuspid valve? Because of the shape, yeah, which looks like has three leaves. Yeah. Then the valve on the left side of the heart, from the left atrium into the left ventricle, is called the mitral valve. But both tricuspid and mitral valve are basically, we call them as atrioventricular valve, the valve that limit or bordering the flow between atria and ventricles. So here, because it has different structure, in A, this is tricuspid valve from the right atrium to the right ventricle, consisting of three cusps of the valve. Yeah. Semiluminar valve built out of the three pocket-like cusps. The schematic closure and opening of the semiluminar valve. We compare with this one. Okay, so as we have mentioned that the heart is basically an electromechanical device, the electromechanical organ, because this motion, this heart contraction, it has certain rhythm, because it has its own also electrical system that excite the stimulus from locally there in the heart. So this is different, for example, with the muscles in the limbs, the upper limb and lower limbs, where we are conscious of them and we can move them based on our command from our brain, based on the command from our brain. So the heart, on the other hand, works autonomously and in a rhythm because it has its own electrical network. The electrical excitation here is drawn in this figure. Electrical excitation of the heart through a conductive system consisting of specialized cardiac muscle cells. So this arrows indicates the propagation direction of excitation in the course of the cardiac cycle. Yeah, so the electrical excitation starts from this node called as the sinoatrial node. The sinoatrial node is located on more or less on the upper part of the right atrium. Okay, then it sends electrical signal into the atrioventricular node through this internodal fiber. Okay, to the internodal fiber. So to the right and to the middle, and then also to the left side of the heart. The atrioventricular node is located between the atrium and the physical. Okay. Then it is branching from this atrioventricular node in what we call as a bundle of His. This is from after bundle of His, it is branching to the right side of the heart and the left side of the heart. Yeah, to both the ventricles. Yeah, and then the electrical excitation is spread throughout the ventricular space using this at the bottom of the ventricular space using these Purkinje fibers. Yeah. So this is how the electrical excitation occurs. It starts from the sinoatrial node, goes into the atrioventricular node through the internodal fiber, then go to the His bundle of His, after that spreading through the Purkinje fiber. Okay, so as we mentioned that the heart basically is functioning as a double pump. So the oxygenated blood from the body, yeah, which contains carbon dioxide mostly, and other products of metabolism from the cells of the body, somatic cells, will goes to the right atrium of the heart, then goes to the right ventricle of the heart before being pumped into the pulmonary artery, goes to the right and left side of the lung. Yeah, like this. This is the heart. So from the upper part and lower part to the atrium first. Oxygenated blood, oxygen-rich blood, come from the lung, from the vein, pulmonary vein, yeah, here from the left and right, goes first to the left side atrium, and then go to the left ventricle before being pumped through the aorta, and then distributed all along the body. Okay, so this is double pump of the heart. The right side pumping blood through the pulmonary circulation. The left side pumps blood through the systemic circulation. The systemic refers to the rest of the body besides lung, where it distributes the oxygen-rich blood, while pulmonary circulation give transfer or transport the oxygen-poor blood, the carbon dioxide-rich blood into the lung for exchanging gases with the surrounding atmosphere. Closed atrioventricular valve and open semiluminal valve indicate blood ejection phase during systole. Okay, so this is the closed atrioventricular valve. So when it is closed, then the semiluminal valve is the one that connects the vesicle with the either lung or the aorta. Okay, so this is where blood is being ejected or being pumped from the ventricle. This is what we call as the systole stage of the blood circulation. Now, this is the connection. These figures shows the connection between the electrical excitation of the heart that we have discussed in the previous slide, from the sinoatrial node until the Purkinje fibers, and its correlation with the mechanical pumping of the heart. So this shows the single cycle excitation of the heart. And this is the famous one single cycle. Usually then will form the famous ECG graph, ECG waveform. We call it as PQRST. So P in the upper part, then Q, P, Q, Q in the lower part, bottom part before it jump, R, the peak, S, R, then S here, then T, the peak before the one cycle ends. So here is the beginning, what we call as the inflow phase. When it starts to affect this, it is in the relaxed state. Afterwards, then blood flows. Blood flows to the atrium at the in the upper chambers, the atria, both atria, from the pulmonary circulation as well as the systemic circulation. So this is the ventricles are not yet excited, only the atria. Then it forms this P, the half of the P wave. So after the second half, so the P wave is completed. So this is also still completing the inflow phase of the atria. So blood is fulfilling the two chambers of the atrium, the upper part of the heart. Then goes to the Q, yeah, where the electrical excitation goes into the bundle of his, and then starts going towards the branches in the ventricles. Here you see the dipole now is located here. It means that this is very the electrical field is located towards this downward. So this will then makes the blood to fill the both chambers, both atrium, from the atrium, yeah, what we call as the isometric contraction. Okay, this is the R, where it starts also to be being pumped. Yeah, the blood is start to be pumped of the ventricles, one to the lung, the other one to the rest of the body. Then the R is completed, where isovolumetric contraction is completed. Then afterwards, ejection, and where now the dipole is shifting towards the left ventricles. And then this is where the T wave occurs. After that, it starts another relaxation before it starts another cycle. So, and this is the relationship of the PQRST ECG waveform with PCG. PCG is phonocardiogram. Yeah, the signal of the heart that is captured through stethoscope, the sound signal of the heart. And then we measure also the blood pressure. Here, the systole, the blood pressure, the systole and diastole. So you see here at PQRST, the systole occurs exactly at starts, yeah, happen at R. So when we look here at R is where it starts this isovolumetric contraction of the ventricles, starting to pump the heart from both ventricles, one to the lung, the other one to the rest of the body. Then, after it finishes the QRS cycle here, before the T is where it starts the relaxation. Yeah, the T start the ejection. All the blood is completely ejected from the ventricles, and then starting relaxation before initiating the next cycle. So that is where we have this dichrotic notch, and then the diastole occurs. Yeah. So at the PQRST, RST, where this is the ejection, the systole blood pressure. This is where the semiluminar valves are open, yes, especially from the time of B. So B here, if we look here, more or less related to just after S wave. Semiluminar valve is the valve that goes to the arteries. So one is to the pulmonary arteries, the other one to the aorta. Then afterwards, the atrioventricular valve is open. This is where the blood starts to flow again from atrium into the ventricles. Yeah, so this blood is completely pumped, ejected, and then blood starts to flow again from the upper chamber, from the atrium into the ventricles at this diastole stage, and where that occurs usually after the repolarization, after the T wave, yeah, until the P wave. Yeah, then that occurs, the filling of the ventricles from the atria by the blood will be causing the second heart sound. While the first heart sound, the larger amplitude sound, is heard when when the ejection occurs. So the dub dub, yeah. So when we try to hear this using stethoscope, this is what makes the dub dub sounds. Okay, is it clear? Any questions so far? Okay, if not, then we go to the so this the function of the heart, yeah, and the related physiological condition which form these various biosignal, ECG, PCG, phonocardiogram, blood pressure, and also volumetric volume, yeah, the blood volume. Now let's study the other part, the circulatory system, which is the blood distribution network transferring the gases, nutrients, and other liquid substances to and from the cells. So the function of the circulatory systems include transportation of diverse substances, yeah, respiratory, nutritive, or excretory, protection against abnormality like clotting and immune mechanism. So our immune system also depends on the circulatory system. Hormonal and body temperature regulation, yeah, where hormones are being distributed to the from the glands, from the corresponding gland into the appropriate organ to regulate their function. Cardiovascular system is used here for blood transport, yeah, where it carries red blood cell, white blood cell, and many other things, nutrients. But there is also lymphatic system. This is the transport interstitial fluid towards the heart and empty it into the subclavian veins. The lymphatic system is the intertissue, yeah, fluid, the fluid between the tissues. It removes interstitial fluid to maintain the fluid balance in the body. Then transport also fats from the small intestine to the blood and provides immunological defense. So here is basically the illustration of how circulatory system occurs, the function. Yeah? As we mentioned, the heart plays a main role here, big role, because it pumps the heart in the pulmonary circulation, the oxygen poor blood, yeah, or CO2 rich blood into the lung for exchanging gas with the surrounding air, with the atmosphere, and then the oxygen rich blood is being pumped by the heart towards the rest of the body, what we call as the systemic circulation. First to the aorta, and then being branches into the arteries, the upper artery, lower artery, then to the smooth muscles, to the arterial, and then to the capillary system, yeah, the systemic capillary system. Then it captures again to this arteriole, back to vein, yeah, and then back to the right artery. Same thing happens with the pulmonary circulation, similar. So the thickness of the arrows here indicates the level of the blood pressure. So you see here that the highest blood pressure occurs from the left side of the heart, pumping the strongest to the rest of the body. Yeah, so that is why if we look again at the ECG signal, here the dipole, the heart is inclined like this. Yeah, so in the current dipole also from pointing to the middle, to the bottom middle here, will shift towards the left. Yeah, so that this left part will pump the strongest, yeah, to pump the blood from the left ventricles to the rest of the body through the systemic circulation. So in the what signals can be captured, yeah, from the circulatory system? So circulatory system of human with indicated main arteries followed by signal establishment. So here we have the arteries and the veins. And the arteries distribute the oxygen-rich blood, The veins distribute the oxygen-poor blood or the carbon dioxide-rich blood. So, for example, here at the elastic carotid, musculobrachial here, musculoradial, musculofemoral, different biosignal can be captured. Usually they are because of the branching. They're like this. This is the model. The model as asymmetric T tube. Yeah, asymmetric because one has shorter branch, the other one is longer branch, which schematically indicated arterial stiffness and peripheral resistance RT. So this indicates peripheral resistance RT, qualitatively represented by the line thickness. Black color for young person and black and gray color for elderly person. So when you are, especially for you, because you are mostly young, your blood vessel taken only at the end part, yeah, like this. But older people, like from my age, then even at the beginning of the branching, the blood vessel starts already to be thickened. This will create a higher fluid resistance, yeah, because of the increased arterial stiffness. The upper and lower part of the body correspond to the upper, shorter and lower longer limb of the T tube. Yeah, here this represents because the upper part represent the upper part of the body which has a shorter limb, while the lower part here represent the longer limb. Yeah. The aortic valve here, this is the aortic valve coming directly from the left ventricle of the heart. This serves as the input, while both incompletely occluded ends, this one, with the total peripheral resistance RT, yeah, this one RT, and the passage from the elastic aorta to the muscular arteries, denoted as the filled triangle here. This is the filled arteries, serve as the reflecting sides. Okay. So here we will have the incident wave, the reflective wave. Yeah, so in the young, it is still like this, yeah, because there is the volume. Young people like you can pump more volume of blood. Yeah. While in the older people, because of the arteries become what do you call it? The blood vessels become stiffer, and also there is the peripheral resistance is higher. You see here that less volume is being pumped. Yeah. So this can be measured by the incident wave and reflective wave delta t, the time difference. So from here, there we can approximate properties of the arterial circulatory system and the aorta, arterioles, and the capillaries. The vessel diameters 2R and the mean blood flow velocity here. So when the blood reach the capillaries, the mean blood flow is already very small, because the diameter is also small. So the blood velocity also decreases. So it B indicate here the systolic blood pressure, PS, yeah, at the different part of the blood vessels. At the it is highest at the arteries, the systolic blood pressure, then goes down at the arterioles and capillaries. From there we can, this is all the propagation velocity of the pulse wave coming from the pumping of the heart. Yeah, this is in meter per second, this is in kilopascal, this is in millimeter mercury. This one is the diameter of the vessels at aorta, arteries, arterioles, and capillary, and this is the corresponding blood flow velocity in centimeter per second. So therefore, in circulatory system like this, yeah, in the blood vessel, the outermost usually consists of collagen fiber. Middle layer here, the brighter color, consists of elastin, smooth muscle or elastin fiber. The innermost layer here contain the endothelial cell. So it has layers, yeah, in this blood vessel. And then this functions to distribute the red blood cells and also other parts of blood. So this graph indicates the diameter, the diameter here, and the pressure. The pressure. When the diameter high at the aorta, the pressure is very high, but when the diameter is small at the arterioles and capillaries, the pressure is also small. And when we see here in the young age, there is a clear difference when there is of the region where elastin is the most, and this is the region where collagen fibers is dominant. But when people starts to age, because here it is more elastic compared to collagen. The elastin is more elastic. But when people age, then the elasticity will decrease. So here you see that it tends to have less difference in between the different diameters, even at small diameters at the capillaries and the big diameter at the arteries. So this is the arterial behavior. Large elastic arteries act as a cushioning reservoir or wind castle. So the pressure pulse is absorbed by the elastic arterial wall during systole after the opening of aortic valve. This is the stored energy will be pushing from the elastic wall used to drive the blood during the diastole after the closure of the aortic valve. Now this is the system, the steady flow system of the circulatory system. This is very much of fluid mechanics. The schematic representation showing how the concept of vascular complex longitudinal impedance. The longitudinal impedance is calculated from the pressure difference between two different points within the vessel divided by Q. Q here is the flow, the blood flow volume, if I'm not mistaken. And this is at when the blood vessel is make circumference, make a curve, and will be then transmural pressure in the PT in the arterial wall. So then we can calculate using Poiseuille equation. The Q, the blood flow volume, as average blood flow velocity times the cross-section area of the blood vessel, which is pi times r squared. So from here, pi, this becomes the dynamic viscosity of the fluid here. Sorry, the average blood velocity here can be calculated from the pressure difference, the dynamic velocity, and the length of the vessel. Okay. If we simplify, from here, this P1 and P2 become P1 and P2, and the other parameters becomes R, where R becomes the fluid resistance or vascular or longitudinal resistance, where R equals to 8 mu L, from here, 8 mu L divided by pi R to the power of 4. Schematic pulse wave propagation with the heart rate Fc along the artery with velocity V, without considering reflection and actual length of the arterial tree with respect to the propagation wavelength lambda. So lambda here, the wave length of the propagation is equals to the velocity divided by Fc, the heart rate. The parabolic profile here of the blood flow is because in the wall, basically it has very small velocity, or almost zero. The highest velocity is at the middle. So the pulse propagation is calculated as V equals to lambda times F. The V is time spatial characteristics of the pulse wave travel, lambda is the oscillation wavelength, and F the cardiac cycle. And most of the time, usually it is 1 hertz, 1 beat per second. So here is the cardiac blood pressure with heart rate FC. So the period is 1 over FC for one single blood pulses. Yeah, FC here in the aorta, multicircular align over time. This is the pressure, the systolic pressure, PS, and then this one is the PD, the diastolic pressure. The waveform looks like this, yeah, for the blood pressure. So the blood pressure comes from this modeling, blood pressure waveform. So because of the narrowing arterial tree, so this is how it looks like. Then we can measure this blood flow measurement. For example, we can put the sensor here in A using echocardiographic, yeah, to measure the using ultrasound sensor at the one of the vein here, or using barocardiogram measurement, measuring blood pressure simultaneously. So this is we can use the what do you call it, like pulse oximeter. Yeah. So you see here from time perspective, the brachial side here will occur earlier compared to the finger side, okay, in time. So the finger side is delayed, and because of the time needed for blood to flow from the brachial side into the finger. So this is a spatial and temporal derivative. The delay is about 30 milliseconds. So this is the P, then the delta P, the gradient basically. This is the dp/dt. Yeah, this is the dp/dt. Using optoplethysmogram, yeah, or pulse oximeter to be short, in young and old pulsative flow, we can measure. This is the young and this is the old, and then this is the systolic pulsatil component. Yeah, and this is systole, and this is the diastole component, and this is what we will measure in optoplethysmogram. This is for the young, and this is for the old people. The waveform, how they will look like. So measuring between the difference between measuring in brachial, yeah, using barocardiogram, and barocardiogram in also in fingers, yeah, but using barocardiogram, so it means using a pressure meter, using, for example, piezoelectric. Later you will, or compared to using optoplethysmograph. The upper part is measuring the mechanical wave, and the lower part measuring the optical pulse. Okay, so I think that will be all for today.